welcome everybody to chapter 2 of MCAT General Biology. When I was taking notes for this, I wrote a bunch of curse words in my notes because it's really annoying. There's a lot to cover and things get really confusing. And so I'm not gonna tell you what was confusing to me because my hopes is that at the end of this, nothing's confusing to you. But if something's confusing, let me know because it gets really, really messy. So let's begin. So we all know DNA looks like this, right? It's like a double helix and whatever. And these are made up of two strands, right? So one strand will look like this. You're gonna have a nucleotide base connected to a 5-membered sugar and then a phosphate group. And you're gonna have a bunch of these, right? So let me draw one more. And then these will be connected and it just keeps going, right? So one strand will look like this. The other strand is gonna be the same thing, but flipped. And then so on, right? And one individual component, like with the nitrogenous base, the phosphate group, and the sugar, we call these nucleotides with the T. But if we're just talking about this, like the nitrogenous base and the sugar, just those two, we call it nucleoside with an S. So make sure you can distinguish these two. Now, I'm talking about DNA, but there's also RNA. The main difference between DNA and RNA involves this 5-membered sugar, right? So let me draw it better. Okay, so this right here is ribose. This is the one that's involved in RNA, which is ribonucleic acid. But when we're talking about DNA, which is deoxyribonucleic acid, we're dealing with a deoxyribose. Deoxy just means removing an oxygen, or in this case, an OH. So we're removing this one right here, okay? This right here would be your deoxyribose. And it's specifically this OH. So this is carbon 1, 2, 3, 4, and 5. We're removing the OH from the 2 position. Now for this part, the N, the nitrogenous base, there's 2 categories of them. We have purines and pyrimidines. So for purines, your general structure will look like this. This is the base structure, right? But there's 2 types of purines. We have adenine and guanine. So I'm gonna draw the first one. Yeah, that's good. Okay, this is one of them, and then the other one will look like this. Okay. So this right here is adenine, and this is guanine. A and G. Now for pyrimidines, pyrimidine is a structure in itself, right? So we know we have, what is this called? It's called a benzene, right? If you replace one of the carbons with a nitrogen, you still have your 6-membered ring. This is called pyridine. And then look, we're going for pyrimidine. Whenever you have an im, that just refers to an extra nitrogen. So pyrimidine will be a pyridine with another nitrogen. So this and then whatever, right? So it's a benzene with 2 nitrogens. So that's your general structure of a pyrimidine. It's gonna involve this larger ring, okay? The purines have 2 rings connected, whereas pyrimidines are just a single ring. So let me draw them out. Okay, this is one of them. And then, how many pyrimidines do we have? There's 3, okay? So this is for DNA right here, right? We have adenine, guanine, and then the one with the NH2 is cytosine, and this is thymine. But there's one more base, which looks identical to thymine, but it's gonna be the one that's used for RNA, right? So this is gonna be called uracil. The only difference between thymine and uracil is that thymine has an extra methyl right here, or CH3, whereas uracil does not. Hold on, missing something. Okay. So remember, thymine is for DNA and uracil is for RNA. Now, I drew this right here, right? Basically, like your two nitrogenous bases are going to interact or form bonds with each other. And the way we know what forms bonds with what is using something called Chargaff's rules. I know this is basic, but trust me, I'm gonna make it confusing after this, okay? So Chargaff's rules tells us that adenine bonds to thymine and then guanine bonds to cytosine. Why do I have 2 and 3 lines here? This refers to number of hydrogen bonds between the two bases. But we're always told that A, has 2 hydrogen bonds with T and G, triple bonds with C. But do we know what that actually looks like? So I'm gonna do the first one, adenine and thymine. Give me a second, I have to draw this again. Okay, so this is adenine, and then thymine is gonna be like flipped. I can't draw these properly, hold on. Well, I don't know how to draw this shit. It's like, whatever. Don't worry about the rest. The main aspect is this stuff right here, right? Here, let me just continue it. Anyways, okay. Sorry, this is really bad. But anyways, this is adenine and this is thymine. Here's what happens, right? We're talking about H bonds. What is an H bond? This is, think back to Gen Chem. Let's say you have a molecule of water, and then you have another one, okay? An H bond is just going to be a hydrogen intermolecularly bonding to either nitrogen, oxygen, or fluorine. These are our 3 most electronegative atoms. So we have 2 molecules of water. This oxygen, it has 3 lone pairs, right? And it encounters this other water molecule, which has an H, a hydrogen. So this one of the free lone pairs is going to facilitate a intermolecular bond with the other H. And so that's like what's happening over here, okay? Right here, we have an oxygen with the lone pair. And let me draw this like this. So the NH2. It's gonna facilitate H bond like that. Same thing over here, this nitrogen has a lone pair. It's gonna facilitate this H bond. I don't know if I want to draw the other one. Here, let me just draw the other one real quick, the guanine and cytosine. And then I'm gonna make it confusing right after that. This is guanine, and then cytosine will be... If at any point you get confused, just let me know. Or if I'm like making it messy or whatever. Okay, this is rest of it cytosine. So look, for cytosine and guanine, here we have a lone pair here, facilitates this H bond. Nitrogen lone pair facilitates this H bond. And then lone pair with oxygen facilitates this H bond. That's where we get the 2 and the 3 from. But there's exceptions to this rule. So I'm gonna bring up a organic chemistry term real quick. We call it tautomerization. I'm gonna explain this the long and hard way, and then I'll make it easy afterwards. So tautomerization, there's 2 types. One, we call it keto-enol tautomerization. This is a keto, like ketone, and enol because alkene and alcohol, enol, right, E-N-O-L. And really, really quick, I'm gonna go over the mechanism. We're gonna be like better at this in organic chemistry chapter, but really quick, what happens is you start with a ketone, and then you're gonna have a base come in, and these, there's 3 hydrogens here. The base is gonna take one of the hydrogens and deprotonate it. What do you get after that? You get this negative right here. And then this is gonna come in, form a double bond, kick this up, and you get O- with this alkene. And then this OH, it was an OH, but it gained that hydrogen, so now it's water. So we have H2O. It's gonna protonate this oxygen, and you're gonna make this, okay? Once again, it's whatever. Just know that you can go from a ketone to an enol. The other type of tautomerization is called imine-enamine. So in imine, I-M-I-N-E, this is an amine, but with a double bond to a nitrogen, so it looks like that. And then enamine is going to be alkene within amine right here. And these are not like proper resonance because it involves movement of a hydrogen. So it's not just straight up resonance structures. They're actual different structures. But this is called tautomerization when you can like switch from one form to the other. So look at our bases right here. We have a keto, 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 and then this can be called like an enamine because we have an alkene and amine. So these structures can also do that. They can tautomerize. These structures I drew right now are the normal structures. This is what they're normally found as like 99.9% of the time. But tautomerization can occur. And what happens if tautomerization occurs? I'm gonna tautomerize this thymine right here. So what we're gonna do is, this is a keto, right? I'm gonna make an enol. So I'm gonna make this like that. And then there's a double bond here, and this H goes away. Now let's try to do the same double bond stuff that we were doing. There's an H right here. Okay. First of all, there was an H bond going on over here, but it's gone. There's no hydrogens on either of them. So that's, there's nothing there. There's nothing down here either. So what about this? First of all, if this does work, it's only a single H bond, which is not sufficient. But is there even an H bond there? Okay, this oxygen has a lone pair, and this nitrogen has a lone pair. Can either of them form an H bond? Yes or no, and why? Okay, so the answer is no, and it does not have to do anything with the fact that both of them have hydrogens. It's because, look, we need a free lone pair, right? Neither of these lone pairs are free. Why? Because look, this lone pair, oxygen, is right next to a double bond, so you can have resonance and all that stuff. Same with this nitrogen, you can have resonance like forming a double bond, and then this kicks up, forms a lone pair there. And so resonance is like a hybrid thing. So your lone pairs are not fully there. They're in a constant state between lone pair and double bond, lone pair, double bond. So if the lone pair is not fully there, then you can't use that to form an H bond. Now, why does something like this work? There's a lone pair and you still have like resonance as possible. It's because if you did have like resonance occur, like this double bond became a lone pair, you're just adding another lone pair, but this lone pair that was there to begin with is not moving anywhere, it's still there, right? So now, what is this tautomerized thymine going to bond to or pair with? Okay, I'm going to, here, let me, you know what, I'm gonna just redraw it. But any guesses like what this tautomerized thymine will bond to now? Oh my God. Here, it's gonna be guanine. Whatever, I'm not drawing the rest of guanine. I'm just gonna draw the main part. Okay. Look at this right here. This is still thymine, it's just the tautomer, right? So it's T, hold on. It's T, but in the enol form, or the tautomerized state. This is regular guanine. Now look what can happen. Lone pair, H bond, lone pair, H bond, lone pair, H bond. So look at that, thymine is forming 3 bonds. What the hell? Thymine normally forms 2 bonds with adenine, but now it's forming 3 bonds with guanine. So basically, I'm gonna make this easy. I just explained the whole way it works, just in case anybody's curious. But here's all you have to know. A purine, actually, wait, before I get there, if I tautomerize guanine, what do you think it's gonna bond to? And like how many bonds? I wanna see if like y'all pick up a pattern here. I mean, there's not really a pattern to be seen yet, because I only showed one example, but I'm just wondering like what the thought process is. So we tautomerize thymine, and now it bonds to G with 3 bonds. But if I tautomerize like guanine, what is it going to pair with and how many bonds?
It will pair with adenine and it will form 2 bonds.
Okay, so we got adenine and 2 bonds, right? Is that what you said?
Yes.
Okay. So unfortunately, that is incorrect. Here's what you're gonna have to do, right? Just know that purines must bond to pyrimidines, okay? So normally, whatever, G bonds to C, but if it gets tautomerized, it's just gonna be the other pyrimidine, which is gonna be thymine, and it'll be 2 bonds, right? look what we did. We had T double bonding to A, but we tautomerized it. Now it moves down here and triple bonds to G. So that pattern is going to continue. And I'll just write all of them, right? I'm going to show like the tautomerized base pairs, BPs, right? So the first base is the one that gets tautomerized. So if you tautomerize T, it's going to triple bond to G. If you tautomerize adenine, it's going to triple bond to C. If you tautomerize guanine, it's going to double bond to thymine. And then if you tautomerize C, it's going to double bond to A. And I know this looks weird, like A triple bond to C and C double bond to A, but it's just like, depends on what gets tautomerized. But the main takeaway from this, I don't think you have to know like in depth like the number of bonds, but I've seen questions a couple times where it asks you if thymine got tautomerized to its enol state, what base is it going to pair with? It's just going to be the other purine in that case, right? So thymine would bond to guanine. All right, now purines and pyrimidines, how do I remember what is what? So first of all, I would really recommend being able to draw them out like I did, because you never know, like maybe they'll ask you how many nitrogens does guanine have? And like, look, adenine has like, hold on. Actually, it doesn't really matter. But anyways, like, they might just ask you some random like structural question, and it's good to be able to know the differences between all of them. But just being able to recognize them, I guess, is sufficient. But if you really want to maximize your score, it's good to be able to draw them. Now, how do I remember purines and pyrimidines? Does anybody have a cat? So there's this brand called Purina, and I remember in freshman year, like 9th grade pre-AP biology, this one kid, he just yelled out, a good Purina bag. So it's like adenine, guanine, Purina, like purines. And then this one, he said cool thick pyramids. So I always like, that's stuck in my brain, cool thick. So cytosine, thymine, and pyrimidines. And then uracil is just uracil. It's like, we call this kind of like an analog. So like it's basically a copy of thymine. But once again, the difference is that it doesn't have the extra methyl. Now, as for being able to recognize these, let's say you didn't know how to draw them or whatever, and you ask a question like, which of these is adenine, which of these is guanine? Here's how I remember that. For this one, I look at the top element is what's different, right? We have a nitrogen here versus an oxygen. I just put these together, like I have the word go and. These are both like 2-letter words, right? A-O and G-N is not a word, so that's kind of how I remember, like G goes with O and A goes with N. Now for these, the same thing. I look at thymine. Thymine has an O, so like, it's right over here. T-O is a word. C-N is not a word, but like C-O is also not a word. So I just remember like this, like T has the one with the oxygen on top. And then C will be the opposite. And then uracil, once again, is an analog, is a copy of thymine without the extra methyl. Confusing or not? Okay. Okay, let me get rid of this one. Actually,
there's also cut the pie for a mnemonic.
I knew you were gonna say that. Yeah, that's like another mnemonic. I just go with mine, but like, there's definitely. Yeah. I think the other one is pure as gold. That's like a very common one. Yeah. Okay. Now, let's talk about like chromosomes, right? We talk about chromosomes a lot, or we just hear that word a lot. But before I get into chromosomes, let's like build up to it. So we have our DNA that looks like this, right? It just stands. But there's something you can introduce called a histone protein, right? So you're gonna have your DNA, whatever, and it's gonna like kind of wrap around this and then continue. This is called a histone. It's a protein, right? A histone involves lysine residues. Lysine, the residue will look like this. Positively charged residue. DNA is negatively charged. Why? Because of the phosphate group. So this is like a minus, right? So whenever you introduce a histone protein complex, it's going to basically bind to the DNA and kind of like condense it in a way, okay? Okay, I'll continue with this. If you have a bunch of these histone DNA complexes, I'm gonna draw them like this. By the way, this like complex with all of this is called a nucleosome. Okay. If you have like a bunch of these, this is the DNA, okay? This is gonna be called chromatin. So it's like a less full version of a chromosome. Chromosome is like fully condensed. Chromatin is not, it's still condensed, but not fully condensed. And it's more like a segment rather than the entire thing, right? So chromatin, there's two types of chromatin. We have heterochromatin and euchromatin. For heterochromatin, the the histones and all that will be more tightly interacting with the DNA. So for heterochromatin, think like tight. And if DNA is really tightly connected to histone, what's going to happen is going to be inactive. Mainly, like that area that's connected. Now, euchromatin is the opposite. It's going to be loosely connected. And the DNA will be more active. So really quick, if I have, there's something called an HDAC, okay? This is histone deacetylase. So if I have a histone deacetylase inhibitor, it's a bunch of things you have to connect. If I have a histone deacetylase inhibitor, what's gonna happen to the DNA? Is it gonna become more like heterochromatin or euchromatin? This is, I see this definitely show up in passages, and they'll literally ask you, like, if an HDAC inhibitor was introduced, what happens to the DNA? So what are your thoughts on this? If I introduce a histone deacetylase inhibitor, what happens to DNA?
More active.
Okay, so more active. Let me explain how to work this out. So look, let's break this up, right? Histone deacetylase. What does acetylase do? Acetylase will acetylate the histone. Before I get into this, once again, right, histone is lysine positively charged and DNA is negative, right? So if you acetylate histone, acetyl will look like this, something like this, right? It's like an acetate ion. This is negative as well. So if you introduce this to the lysine, it's going to displace this negative DNA, and now your DNA is no longer connected to the histone, okay? So that's what that is. So histone deacetylase. What does acetylase do? It acetylates it. What is acetylase going to end up doing? It's going to free the DNA. It's going to be loose and active. Now, what does deacetylase do? It prevents acetate from bonding to the lysine. And so if you have just a deacetylase, your DNA is going to be more like heterochromatin, where it's tight because it's bonded to the histone. Now, if you have a histone deacetylase inhibitor, you're just inhibiting the very last thing I said. You're inhibiting DNA from basically being tightly connected to histone. And so you're going to have a euchromatin type DNA, which is more active and loosely connected. All right, now, let's go a step further. Chromosomes. So when you have like a bunch of these chromosomes all together, you're going to have a chromosome. Now, this shit is really confusing, okay? We normally see chromosomes like this, but this is also a chromosome. I'll explain this, but just keep this in mind. Both of these are considered chromosomes. This is also called a chromatid, but don't worry, I'll explain that. But first, let's talk about how many chromosomes we have. How many chromosomes do we have, like in our normal somatic cells? So we hear two numbers, 23 and 46, right? When we talk about like our normal cells, like everything that's not our sex cells, we have 46. And why? Look, chromosome number 1, we have 2 copies, right? One from the mother and one from the father. And then, same with 2, they just look different, whatever. Here. But here's the thing, okay? These both are chromosomes. Let's say, this chromosome 1 codes for like eye color, hair color, and height. Both of these will code for eye color, hair color, and height. But since one is from the mom, one is from the dad, they might code for different things. This might code for blue eyes, black hair, and like short height. This one might code for like green eyes, blonde hair, and medium height, okay? So we call these 2 chromosomes in the same set, we call them homologous chromosomes, or just homologs. This is a very important term to know for meiosis when we talk about that, okay? But we have 23 sets of chromosomes. This 23rd set is going to be either XX or XY. XX is if you're female, and XY is if you're male, okay? So look, we have 23 sets of chromosomes, but 2 copies of chromosomes in each. That's where we get this from. We always hear about 2N and 1N, right? So our somatic cells, remember, somatic just means the body cells, everything that's not sex cells. These are going to be 2N, which is called diploid. Write this backwards. Diploid, which is 2N. And sex cells, or gametes, are going to be haploid, which is 1N. What is N? N just means how many sets. We have 23 sets of chromosomes. Somatic cells have 2 copies per set of chromosomes. That's where we get the 46 from, okay? 46 chromosomes, somatic cells. Our sex cells only going to have 1 copy of each chromosome. So 1 times 23 is 23. And why do sex cells only carry half? It's because 2 sex cells come together and they basically make a new organism, right? And they reestablish that 46 chromosome number. Okay, so back to this, right? Once again, I'll explain it again when we get to it, but this is a chromatid / chromosome. If you have 2 of them, this is still a chromosome made up of 2 sister chromatids. But look, both sister chromatids are identical. So if you're dealing with something like this, you just have double the amount of DNA, but they come together to form like an X structure. But if you had this, you're still coding for the exact same stuff. You just have half the amount of DNA. So did that make sense? Like, was that confusing or not? Because like, I don't know, when we learn this stuff, like terminology gets really messed up. Okay, if anything is confusing, just let me know. Give me 1 second. I just want to make sure I didn't leave anything. Yeah, okay. So now, let's talk about the cell cycle. Let me erase all this.
I had a quick question.
Yes.
So both are 1 chromosome, but the first one is composed of 2 sister chromatids, and the second one is half of the DNA, right?
Yeah. Yeah. Half of the DNA. Basically, like still has all the stuff that chromosome 1 has. This also has all the stuff that chromosome 1 has, but it's just doubled. And the reason we have chromosomes that are shaped like this is mainly for replication, where like when you make 2 cells, it's gonna cut in half, and 1 cell will have this, and the other will have the exact same thing. But they're both coding for the exact same thing. This just has double the amount. But it doesn't make any different. It just has extra that it can give away.
That makes sense. Thank you.
Uh-huh. So now, cell cycle. Okay. So our normal cell cycle will look like that, okay? We have G1, S, G2, and M. I tried to draw it in such a way that you can see that G1 is the largest phase or the longest, and then, M is the shortest, but out of G1, S, and G2, G2 is the shortest phase. Now, G1, S, and G2 collectively refer to interphase. And then M, mitosis, meiosis, this is just called the M phase. Pretty basic. So, G1 and G2, the G stands for growth, and then S refers to synthesis, and M is just like mitosis, meiosis. S is synthesis of DNA, okay? But G1 and G2, they're pretty similar, but there's some slight differences. So G1, can everybody see this, by the way? So I'll make sure I don't go too far. I think it's fine. Okay. So G1 is like the first growth phase. This is where you're gonna have organelles being replicated. Mitochondria is also like very important organelle here because you need lots of energy so that you can undergo DNA, replication, right? You're also developing lots of mRNA for the proteins that will be used during DNA replication. And then it's just overall like growth and all that, right? But there's something over here called P53. This is a tumor suppressor protein, okay? What do tumor and cancer cells do? They continuously rapidly divide until you get like a big, I don't know what you call it, like accumulation of cells, and it's bad, right? So a tumor suppressor protein is going to be active here to make sure that there's no abnormal cell division. And if it notices that this cell does not need to divide, then it will just stop division or replication. And how does that work? Yes, I do this, but there's a little like shunt, if you want to call it that, that leads to something called G0. So a cell that ends up, over here we call it like arrested, it gets arrested in G0. It's just a cell that does not need to divide and has no other place in the cycle, so it just stays in G0 and it's dormant. So yeah, one more thing, like decides if cell needs to divide or replicate, or rep for replicate. Okay, now S.
You said P23 or P33? Sorry.
P53.
Okay, thank you.
Yeah, this is one of those things like you don't have to necessarily know it on your own. You're gonna see it in passages most likely, rather than it just being a standalone question. But it's still good to know. It makes it easier to understand passages. All right, S phase. This is, hold on. Okay, there's checkpoints, right? I'll elaborate on checkpoints, but let me just first draw checkpoints. We have a G1 M checkpoint, and we have a G2 M checkpoint. But don't worry about these for now. They're just checkpoints, right? They see if we need to progress. But I'll elaborate on that after. S phase is DNA replication, okay? I don't like talking about DNA replication. It's just annoying. There's not much you need to know, but there's just like certain like enzymes that you should be aware of. So I'm gonna very, very briefly talk about this. So you have DNA, right? Whatever, and then it opens up like this so that you can create new strands. And over here it's like, I don't know, spinning, spiraling. So DNA usually is like a spiral, right? But it opens up into this is called like a replication fork or replication bubble. The enzyme that does that is helicase. It's gonna open up the DNA into this fork that allows for replication to occur. This is gonna keep occurring like that, right? As it gets to it. Now, as this occurs, for some reason, the other side of the DNA could potentially, we call it supercoiling, where it twists too much. I don't know how that works, but it just could happen. So something that prevents supercoiling is called topoisomerase. So prevents supercoiling. And then helicase, I'm gonna just say open up the helix, okay? Okay, now there's something called primase. Primase just puts these things down called like primers. And primers are like sites that the DNA polymerase can kind of like bind to and then continue replication. I forgot the exact reason why. It can't just automatically start replication. It needs a primer site. But you don't have to know all that. Just know that primase deals with primers. And then I just mentioned another one, right? DNA polymerase. DNA polymerase is the actual thing that makes the new strands and all that, right? And then one more thing, when you open up this DNA, what's preventing it from just reconnecting? We call it annealing, right? Re-annealing. What's preventing it? Literally nothing. And so to prevent that, what our body does is it puts these things called SSBs. These are single-stranded binding proteins. They just like complementarily bind to different nucleotide bases, and so now they can't reconnect. And then you can have DNA replication occur, and as you get to these parts, they just get removed, okay? But yeah, that's pretty much it for DNA replication. Now, okay, on that note of DNA replication, let's be confusing again. So DNA, how does it replicate? Is it, there's three types: conservative, semi-conservative, and dispersed. Which one is DNA?
Semi-conservative?
Yeah, DNA is semi-conservative. But what does that even mean? So, if you have a DNA strand, I'm gonna draw it as lines. when this replicates, what's gonna happen is that these are two new DNAs being built, okay? For one of them, you're gonna have one of these original strands, and the other one also has one original strand. And then the new strand comes in. So why is it called semi-conservative? Because you're partially conserving the original DNA, right? You're conserving one of the original strands, but then the other second strand is just a different one. If it was conservative, it would look like this, where you still have the original DNA molecule, right? So now there's this type of question that I've been asked in like Bio 1 genetics, and surprisingly I saw in the MCAT, which is why I'm explaining this now. And let me write out a question really quick and then I'll explain it, because I don't know how to explain it without showing the question. Okay, this isn't a question, it's just like a scenario, but I'll get to the question part. So we have a cell that was grown in N15. N15 is like nitrogen 15, right? It's just an isotope of nitrogen, so it has a mass of 15. And then it grew in that N15, but then it suddenly switched to N14 media for like 2 cycles. So let me draw this out and I'll explain like what type of questions you could get asked, or like the general question that gets asked, right? So I'm gonna put this blue for N15 and then red for N14, okay? So the cell was grown in N15, so all of its DNA will contain this N15 isotope, right? So original DNA at this point is made up of 2 blue strands, which are consisting of N15. Now DNA is semi-conservative, right? So the new strands will both have one of the original N15, but now they're in a N14 medium, right? And so that means that the new strand that gets made with whatever media is there is going to include N14, okay? So both of them now have N15 and N14, N15 and 14. But this says 2 cycles, okay? So this is 1 cycle. What if this replicates again? Okay, this is our new original strand, right? So we're gonna have one original, which is blue, and then the other original is red. Same thing over here, blue and red. Now, what is the new strand gonna look like? Well, we're still in N14, so the new strand will just be N14, which will be red, like this. So what is our DNA composed of here? Over here, we have N14, N15. It's like a hybrid, right? Mix of both isotopes. And then this one is pure N14. So we can write N14, N14. And so they might ask you, like, what is the DNA made of? And you could just say, like, a hybrid of this and sole, or like, what's it called, pure N14. Another thing is, like, if you, like, try to, like, measure these, like, in a tube, there's, like, 3 bands you could get. The higher band would be N14, like pure N14. The middle one would be a hybrid N14, N15. And then the bottom one would be N15, N15. It's just because as the mass increases, the density increases, right? So N15, pure N15 would just settle to the bottom, whereas pure N14 would be the highest and the hybrid is in the middle. So they might ask you, like, what is your band gonna look like? And then you could say a hybrid, this one, and then like a pure, whatever, N14, N14. Any questions on this one? This is one of those things, like, you don't really get taught. I only know it because my teacher explained it, but like I've never seen anyone explain this one. So I want to make sure that everyone understands this, because I don't know where else you can learn this. Is everybody good with this one?
But the band wouldn't show for N15 and 15, right?
No. Okay, yeah, I just drew this to show examples.
Okay.
But yeah, you just get this right here.
Yeah, okay.
The good question though, that was my fault. Okay. So now we're done with S phase. Let's talk about the G2. So G2 is after DNA was replicated. So it's going to check to make sure that DNA was replicated properly. If DNA was replicated improperly, there's something that can occur called MMR, which is mismatch repair system. So like if you have bases that were like incorrectly paired, then it's just going to fix that up. But what happens if the errors in the DNA replication were super extreme? Then what happens? It's just going to cause apoptosis. Now remember, apoptosis, not that big of a deal. You're just killing a cell, programmed cell death. You're not, look, you have a cell here, you have all these cells around. This cell right here just dies, but everything else around it is perfectly fine. It's whatever. And then like the last thing you can say that G2 does is it like sets up the organelles in their proper position to prepare for like mitosis, meiosis. And then one more thing, like what's it called? Centrosomes. Okay, so I said set up organelles. What the hell? I spelled this so wrong. Okay, set up organelles, and then centrosomes are like we talked about in chapter 1. Centrosomes are like the complex of centrioles. It's like a little organelle that has centriole inside of it, and these are what spindle fibers come out of. And then once again, I'll elaborate this when we actually talk about mitosis, but this is like a pretty important one. So okay, I'm gonna erase this. Before we actually talk about M phase, let me draw what a cell looks like in interphase. So here you have a replicated like centrosome or set of centrioles, and they're just together, they're not like distinct, pieces at this point. You also have this is your nucleus, and this dashed line is your nuclear envelope. You're gonna have these little dots inside. Let me draw a different color. The little dots inside are your nucleoli. What do nucleoli do? Nucleoli are the organelles that produce ribosomes. And then, I'm sorry if you can't see this, but these purple lines, they're like DNA, but they're not condensed into chromosomes forms. They're just like loosely present, right? So this is interphase right here. Now, the cell cycle, or what's it called, mitotic phase, goes like this: PMAT. Prophase, metaphase, anaphase, telophase, okay? So the first one is going to be prophase. In prophase, your centrosomes are going to start like splitting, right? And they're beginning to form these spindle fibers. And then your nuclear envelope, it starts to go away, starts to disintegrate, whatever you want to call it. And your nucleoli, they're gone, okay? No more nucleoli. And now you have proper like chromosome structures. Now, why am I drawing them as X's? Because DNA was replicated, right? Remember what I was telling you? This is still a chromosome, but it's also a chromatid. Now you have two of them, right? For the same one. And so they just come together to form this X structure, but it's just two copies. Remember, double the amount of DNA. Okay, now there's like the phase in between prophase and metaphase called prometaphase. I'm not gonna really draw it because prometaphase is just like a continuation of prophase. So whatever happens in prophase, like it starts there, but then prometaphase is where it like finishes off, right? So this nuclear envelope that's disappearing, it's initiated in prophase, but in prometaphase, it's completely gone. This spindle fiber stuff going on, it starts in it starts in prophase and then finishes in prometaphase. The only thing that I would say that actually occurs and finishes in prophase is your nucleoli being removed, and I guess your chromosomes being formed, like they're condensing. All right, now we have metaphase, which is prophase, and this is metaphase. So now you have your what's it called? Centromeres, centrosomes, whatever. Centrioles and centrosomes. They're moved to opposite ends of the cell. And now your chromosomes are lined up in the center. We call the center the metaphase plate. And then the spindle fibers, they start attaching to the chromosomes. I have a bunch of things to say here. Firstly, what are these spindle fibers? We talked about in chapter 1 the cytoskeleton. Remember the cytoskeleton? We had microtubules, we had intermediate filaments, and then we had microfilaments. So which of these three are our spindle fibers?
Microtubules.
You said microtubules? I think that's what I heard. Yeah, it's microtubules. What's up? Okay.
Microtubules.
Yeah, it's microtubules, right? Microtubules are the spindle fibers. Now, another thing, here's our chromosome again. There's two words we hear a lot, and nobody ever explains this, okay? Centromere and kinetochore. They're pretty much the same thing, right? Centromere is just the region where the two chromatids are connected, and kinetochore is the actual set of proteins that connect the two chromatids together. So centromere is the region, and kinetochore is the actual thing holding them together. So anyways, these spindle fibers will connect to the kinetochores, right? The proteins that are holding the chromatids together. Nothing crazy. And then we have anaphase. Anaphase is going to be our shortest phase in the mitosis-meiosis. So anaphase, your cell is going to like elongate a little bit, and then you have your, okay, look, we had X's here, right? But the X's are made up of these two chromatids, right? Which become chromosomes. So now you're pulling them apart. And, you get the idea. They're being pulled apart by two things. One is our motor proteins. What are our motor proteins? The main ones are kinesin and dynein. So kinesin and dynein are going to be the ones that actually stretch the cell, they pull it apart. That's one way of moving the chromatids apart. And the other is just these spindle fibers themselves are literally pulling on the chromatids or chromosomes to opposite sides. Now, by the way, okay, I drew like, 6 chromosomes here. But look, you're going to have like 46 of these, okay? Like 46 X's. And then when they get split apart, you're going to have 46 up here and 46 up here. That is going to maintain the diploidy, right? The 2N. So just make sure you're aware of that. Okay, anaphase, yeah, once again, just pulls it apart. And then telophase plus cytokinesis. Wait, can you all see the little details I'm drawing on this? It's too much work to get a different color. That's why I'm just leaving it here. Okay, so here, I'm sorry. So look, what I'm trying to show here is that in telophase, we basically are still in a single cell, but we created two distinct sides that will now become two new cells, right? So what happens here? The nuclear envelope reestablishes. Your nucleoli, right? The organelles that produce ribosomes, they come back. And then now you have the sister chromatids, which once again, they're just single pieces of DNA, but they're the same thing. They're on both sides. And we still have 46 here, 46 here. And then, this is telophase, but there's, right after this is cytokinesis, which is going to split them apart. But really quick, telophase goes like this. It starts like this, like an elongated cell, and then it looks like this right here. Then you have whatever stuff here. This is called a cleavage furrow. This little bend right here is called a cleavage furrow. And in animals, we have cleavage furrows. But in plants, since they have a cell wall, they're not able to like really bend like this. So they just have something called a cell plate. But cleavage furrows, anyone remember what cytoskeleton component causes the cleavage furrow? Like what actually bends them? Or what's it called, like, squeezes them like that? You have these three options. Is it microtubule, intermediate filaments, or microfilaments?
Microfilaments.
Yeah, it's going to be microfilaments, which is actin. Actin is going to actually do this. And then, you end up with two new cells. And they're identical, right? Okay, I erased it. But yeah, these are both going to be 2N, okay? Completely the same. But instead of having the X's, they have the single chromatids. Once again, a chromatid is a chromosome. Okay, now, how do we There's also meiosis, which I'll get into right after this, but I want to continue on this stuff I was talking about, the checkpoints, right? So we're going to talk about how do we regulate the cell cycle. This one also, like, I don't know, no one really explains it that well, but let's talk about it. Cell regulation. Okay, so you have these things called cyclins, right? Cyclins are just proteins that get released at different parts of the cycle, okay? And then you have something called CDK. This is cyclin dependent kinase. Okay, let's break this down, right? It's a kinase that's dependent on cyclin, okay? What is a kinase? This is like one of the most important enzymes, if not the most important enzyme that we ever deal with in MCAT. What is a kinase? Why does nobody know anything? Are y'all just not giving answers, or like?
Phosphorylates, right?
Yes. Can you be a little more specific though? It phosphorylates with what? Because we have phosphorylases.
What a phosphate group.
I mean, yes, with the phosphate group, but from where? So we have, we have phosphorylase. I'm so sorry. Okay, I think I heard the right answer on Zoom. But we have two types of phosphorylating enzymes, right? We have phosphorylase and kinase. Both of these will phosphorylate things, okay? But the main distinction is that kinase phosphorylates using ATP. So you're gonna take a phosphate from here, right? You have like a TP means like triphosphate, so you have 3 phosphates here. And then you're going to remove one of these and phosphorylate something. Also, now that we're on this topic, right, this is, it's gonna look like DNA or whatever, right? It's like adenosine, this is your nitrogenous base, adenine, and then deoxyribose, and then 3 phosphates. But really quick, this will help you with a question, not to spoil, but this phosphate that's directly connected to this sugar is the alpha phosphate, this is the beta phosphate, and this is the gamma phosphate. So kinases, they use the gamma phosphate of ATP and phosphorylate something. Kinases can also like form ATP if they take a phosphate group from something and reattach it, right? You have an ADP, they'll reattach it and form ATP. You'll see this in like glycolysis and stuff. But anyways, cyclin-dependent kinase, it's a thing that phosphorylates using ATP and is dependent on these cyclins. And what are cyclins? They're just proteins that are released at different phases. And so the way this works is this circle will be your cyclin, and then your square will be the cyclin-dependent kinase. When these come together, you're gonna form a cyclin and CDK complex, right? Cool, now it's active. It's an active CDK. And then you have, this might be a little too much information, but like, if anything, it will help you with passages, right? So you have this protein called RB, retinoblastoma, and it's attached to a transcription factor. The reason I say this will help you with passages is because I don't know if any of you have done passages, like you see E2F, and then there's something called like, things like FOX3MP or something. These are just transcription factors, okay? The transcription factor that's used just depends on where you're at in the cycle, but I'm gonna write TF for a transcription factor. All right, so we have a retinoblastoma that's connected to a transcription factor. Transcription factor is inactive because it's connected to this protein. So what's gonna happen? Your activated CDK is now going to phosphorylate this guy. What happens when you phosphorylate it? It's going to change its shape. So now it's a phosphorylated retinoblastoma. Now this transcription factor is released. And then it just helps continue the cell cycle, right? You're transcribing the stuff that will be used for different parts of the cycle. Now, I guess an important thing to take away from all this is that we have 2 types of CDK. We have just CDK1 and CDK2. And there's 2 checkpoints, G1M and, holy shit, G1S and G2M. Which one will be used for which? CDK1, will it be used for G1S or G2M? What about this one? Which one will they be used for?
G1S?
So you said CDK1 is G1S? Okay, that's one answer. What about y'all? What do y'all think? What do you think? CDK1 will be G1S or for G2M? This is actually important, trust me. Okay, so yes, you would think that CDK1 is for this one because it's chronological, but it's the opposite. CDK2 is the one that's used for G1S, and then CDK1 is the one that's used for G2M. All right. I'm just gonna leave this for like a couple of seconds in case anyone wants to write it down.
So in your G1, you're starting off haploid, right?
No. Okay. Okay, I know what you're trying to ask. So that's a good question. For mitosis, you're just all diploid, right? Mm-hmm. Here's the difference though. In G1, prior to replication, you're gonna have 46 of these chromatids, which once again are chromosomes. When you replicate, this is a common misconception. People think you go from 2N to 4N. What actually happens, let me draw this again. You have these chromatids. After replication, you're gonna have 2 of these, right? And they form this X shape, right? Mm. And so you're gonna have 46 of these now. So you started with 46 single chromatids, now you have 46 double chromatids or like X-shaped chromosomes. And this is still 2N because you still have 46 chromosomes, right? You still have 2 copies of each chromosome, but the DNA amount is doubled. I know that sounds a little weird, but let me try to find a better way to explain this, okay? So here's chromosome 1 and chromosome 2, right? You're gonna have 1 from your mom and then 1 from your dad. Okay? These 2 are different. They're different things, right? This one codes for like blue eyes, this one codes for green eyes, and then black hair, brown hair, right? But all you're doing in S phase is you're doubling it. So now you're gonna have 2 of these, but these are the exact same thing. You're not really making a new chromosome. It's the same thing, but you're just making like double the copy. If you added like completely new chromosomes, like, I don't know, a third parent somehow, then this would actually increase the number of chromosomes, but you're just doubling the amount of DNA. And so you're not adding any new chromosomes, you're like adding any new different chromosomes. So you're just doubling the amount of DNA. And so in this case, you're still 2N, you're still diploid. Once again, you just have extra DNA. And the purpose of this is so that you can split it apart and give half to the new cells, right? To answer your question?
Yeah. Wait, so in both cases, for G1 and also in S, you're still staying diploid?
Yeah.
Okay.
Throughout the entire thing from mitosis is fully diploid.
Okay.
It's just the DNA content or amount of DNA will change.
Yeah.
All right. So, okay, yeah, what's up?
I just had a quick question about the CDK.
Yeah.
So what activates it? The cyclin?
Yeah.
Or...
So cyclin, they're released. Sorry, continue.
No, that was just my question.
Okay, so cyclins, they're released for whatever reason throughout the cycle. And then the CDK will encounter it. And then when they connect, they form like this complex. This is what will activate the CDK. Because remember, it's a cyclin-dependent kinase. So it depends on the cyclin. So when the cyclin is there, it's good. It can be activated.
Okay, thank you.
No problem. Okay, so this whole G1S, G2 stuff, I'm going to erase it, but just keep it in mind. This is going to be the same for... yeah, what's up?
When we see CDK not be activated.
CDK will not be activated if there's no cyclin. So if a cell does not need to divide, then maybe a cyclin will not be released. And so the CDK is just going to be there, but it's not going to be activated because there's no cyclins available.
What if this is mutation?
Mutation to what? Fuck, bro. Yeah, what's the question? No, you're good.
Maybe a mutation to the DNA that was replicated.
Yes. If are you asking what happened? Okay. Basically, let me know if I'm not answering your question, but if DNA was incorrectly replicated, then I don't think a cyclin will be released because remember this MMR, this mismatch repair system, that stuff has to occur to fix any errors. And then after everything is good, it's actually ready to replicate, like divide in M phase. Then I'm sure the cyclin releases and then CDK will be active. Was that your question?
I think so.
Okay. Are you sure? Just let me know if I didn't answer it.
Yeah, it's okay.
Okay. So this G1S, G2 stuff, it's the same for meiosis, so just remember these, but I'll refer back to them. But like for meiosis, you're still like doubling DNA and all that.
While you're erasing, do you mind just giving a definition again of kinase, please?
Yes. Or does anyone else want to answer that? All right. So,
I believe I said.
Sorry, I was gonna say kinase phosphorylate using ATP. That's pretty much what.
yeah.
Basically, kinases rip a phosphate off of ATP and stick it onto something else.
Yeah, so let me give a good example, right? Let's do. So we're gonna do glycolysis, okay? Glycolysis, the very first reaction is glucose becomes glucose 6-phosphate. It's just a glucose with a phosphate attached on the 6 carbon. And then there's an enzyme called hexokinase. Hexokinase, let me actually write that out. The hexo refers to like 6 carbon sugar. Oh my, this is really slanted, I'm sorry. So anyways, hexokinase, it needs to use ATP. So it uses ATP, and in return, you're gonna get ADP. This is adenosine triphosphate, right? 3 phosphates. This is adenosine diphosphate, 2 phosphates. Where did that 1 phosphate go? It went towards this. And this is the job of a hexokinase. You can't use like a hexophosphorylase. It needs to be a kinase because kinases deal with ATP. And once again, this structure right here, the specific phosphate that gets taken out is the gamma phosphate. This is the alpha phosphate, beta phosphate, and gamma phosphate of the ATP molecule. The structure of ATP just looks like DNA, right? You have your base in the adenine, your sugar, and then this time you have 3 phosphates instead of just 1. All good?
Just another quick question.
Yes.
But the P53, do they do like a DNA repair or what?
Um,
Not sure.
yeah, P53, I'm not sure exactly what it does. It's just like a tumor suppressor, so it somehow signals whether replication needs to continue or not. But once again, you don't have to really know too much about P53, other than that it is a tumor suppressor protein. Because majority of the time, pretty much every time I've seen it was in a passage where they explain it. I just brought it up because we were on that topic, but don't worry too much about that.
Thank you.
No problem. All right, so let's talk about meiosis. Meiosis has 2 parts, meiosis 1 and meiosis 2. So let's start with meiosis 1. Meiosis 2 is the one that's similar to mitosis. But meiosis 1, you're going to start with prophase 1, okay?
Meiosis 1.
Oh, my fault. Thank you. Meiosis 1, you're going to start with prophase 1. All right, back to our chromosomes, right? 1, 2, 3. We have your chromosomes from your mom and your chromosomes from your dad. Now remember what I said, the G1S stuff continues. So now all these are replicated, okay? You're gonna have, instead of this, you're gonna have X-shaped chromosomes for all of them, right? But the difference here is that instead of all 46 of these being given to each cell, it's only gonna be 23. So just keep this in your mind. So prophase 1, right? You're going to, the main thing that happens here is lining up of tetrads. So what are these called again? Like 2 chromosomes in the same set, what are they called? Yeah, homologs or homologous chromosomes. So this is a very, very key feature of prophase 1. You're going to line up, these are homologous chromosomes, right? This might be like chromosome 1 and chromosome 10, okay? They're going to line up, and right here, they're just gonna, what's it called? Synapse, okay? So it's called crossing over or synapsis. Big term. Where's my red? Oh, okay, whatever. This is called crossing over. So look, originally my DNA looked like this, all blue and all purple. When they cross over, Now I'm gonna look like this. You have a piece of purple on this blue chromosome, and you have a piece of blue. So now look at this. All of these chromatids are different. Right? This one is fully blue. This one has blue, mainly blue, but some purple. This one is mainly blue, purple with some blue, and this is fully purple, right? So this alone is pretty good. It creates a lot of diversity, okay? So this is a single crossover episode, but if you have, why did I say episode? Crossover, right? If you have a double crossover, do you know what that's gonna look like? These are very, very, very rare. Double crossover, right? This is a single crossover where you have just the ends are going to exchange. If you have a double crossover, what's gonna happen is the middle segments are going to change. So here's your X and then here's your, sorry, one chromosome, one chromosome. All right, what's gonna happen is like now the middle segment will get information and then this one will change, okay? So, you know, if you get a question asking how does what does a double crossover do? Instead of two ends exchanging information, what's gonna happen is the middle. I forgot how this works, but the middle is going to have the new information, okay? So now, this is prophase 1. Let's do metaphase 1. Okay. Metaphase 1, listen to this. I'm not being specific with the centriole stuff because, you know, it's just like that part is similar to the mitosis. But here's the thing, okay? In mitosis, you had 46 of these, right? 46 chromosomes lined up. Over here, you're gonna have 2 layers of 23, okay? So over here, you're gonna have some of those and then these right here. Remember, 23 of this top layer and 23 of this bottom layer. And then, you know, I'm not drawing it in that much, but like, don't forget like your crossover stuff. These are pretty genetically diverse chromosomes. Following along, everybody good? So we call this like lining up like, okay, also I didn't draw this properly, but like they're like lined up in the center, right? I drew a little space just because I needed to write this, but they're like lined up in the center, just like mitotic metaphase. So we call this like lining up of tetrads. Tetrads are just like the same chromosomes that crossed over. Now, anaphase, same thing, except now you just have your X's pulled apart, right? And remember, there's 23 on top, 23 on the bottom. And then don't forget, you know, they're crossed over and whatever. And then, this is ana 1. And then telophase, right? Telophase and cytokinesis, same thing. Now you're gonna end up, this is telo plus like cyto, okay? So this is gonna have here 23 of these X's and then 23 of these X's. And don't forget, they're genetically diverse. Cool. Now, between telophase 1 and prophase 2, there's no more of that G1S, G2 thing. This G1S interphase happens just before meiosis 1, but not before meiosis 2. There's a period in between meiosis 1 and 2 called interkinesis. And then, you know, we restart prophase, but this time it's prophase 2. So, okay, prophase 2 is going to be really similar to mitosis. But really quick, what do we have here? From after meiosis 1, what do we have? Do we have 1N or 2N? We have diploid or haploid? Don't forget what diploid means. 2N, which is 46, and then 1N equals 23. Which one do we have? Diploid or haploid? Yeah, this is going to be haploid. So look, normally in mitosis, we replicate both of these, right? Both the mother and the father's chromosomes for each of them, and both cells will get the mother and the father's copies. In meiosis, you're only going to get one. You might have the mother's chromosome 1, the mother's chromosome 2, and then the father's chromosome 3 and whatever, or you have some sort of variation of that, right? But you're only getting one. So this is meiosis 1. It's going to end up as haploids. So these are, yeah, haploids. Now, both of these will undergo meiosis 2, but I'm just going to show one of them. All right. So prophase 1, Nothing crazy. You just have your X's and stuff. So sorry, prophase 2. Here, prophase or metaphase 2. Now you're actually lining up these these chromosomes. Over here, we were lining up 2 sets of chromosomes, like tetrads, right? Now we're just lining up the chromosomes. And this time we're gonna have 23, right? And metaphase, we had 46 chromosomes like that. This one we only have 23, because that's all we have. And then anaphase, you know, typical, they're getting pulled apart. And these are the sister chromatids, but the difference between this and mitosis is that these are not identical, right? Don't forget, I keep saying don't forget, you have some differences. You have like your crossing over and all that, right? And then you're gonna end up, for each of these 2 cells, you're gonna make 2 cells, right? So you're gonna end up with 4 cells. So I'll just draw 4, but this came from both of them replicating. And then each of them is gonna have like 23 of these, but they're all gonna be like mad different. Okay? So this is what meiosis does. After meiosis 1 and after meiosis 2, in both cases, you're gonna have 1N or haploid. Okay, now really quickly, there's a term to know that involves this called non-disjunction. Okay, non-disjunction means if you look at like metaphase and anaphase. Yeah, let's do that one. Okay, metaphase and anaphase. You have these chromosomes lined up. Anaphase, you're pulling them apart. If somehow you mistakenly or just erroneously pull up the entire 2 chromosomes, that's non-disjunction. What's gonna happen? Let me draw it out. So you have your X's here. And ideally, you're supposed to end up like this, right? But what could happen is maybe this. So maybe these are good. But look, this third spindle fiber, pull up the entire, both of them, right? This one chilling over here. And this spindle fiber pulled up nothing because this third spindle fiber pulled up all of it. So what's gonna happen? You're gonna have, for every non-disjunction that occurs, you're gonna have an extra chromosome on here, right? So let's say everything was smooth except one spindle fiber pulled up both of them. Now you're gonna end up with a cell. Let's do this. And this one will have like 2, okay? Look, this is 1, 2, 3, 4. This is 1, 2. So this would basically look like, this was mitosis. It would be 46, but an extra chromosome. So 46 plus 1, right? So 47 chromosomes. This one, everything else was good, had 46. But since this chromatid didn't come, it's less than 1 or minus 1, okay? So what we write is 2N plus 1 and 2N minus 1. Because once again, for every non-disjunction that occurs, you add a 1. So we're saying that only 1 non-disjunction occurred. And so this has 47 chromosomes. Everything else is the same, 46, but gained an extra, and this lost an extra, right? So 2N plus 1, 2N minus 1 is for mitosis. If you're dealing with meiosis, where you're ending up with haploid, it's just gonna be N minus 1 and N plus 1. And yes, meiosis can have non-disjunction occur in both meiosis 1 and 2. The more that it occurs, the worse it is, obviously, right? All right, any questions? This is it for cells and all that. After this, we're going to talk about actual male and female reproductive system. And I'm not going to talk about this chromosome stuff, I don't think. Can someone, what's it called? Can someone compare and contrast mitosis and meiosis? Just tell me like some things that are different, or like, tell me like the differences between mitosis and meiosis. Yeah, so mitosis just leads to diploid 2N, and then meiosis leads to 1N. Another thing is meiosis has crossing over, whereas mitosis does not have crossing over. Mitosis does not lead to any genetic diversity. You're just making the same cells over and over again. Whereas meiosis, you're making completely different new cells, right? Those are like the main things. And then non-disjunction, once again, can occur in any of them. Okay,
Could you say
yeah, what's up?
the tetrads too or no? tetrads.
Yeah, what about them?
For mitosis, I think they have chromosomes, and then if there's one, I think it has tetrads. I'm not sure.
We technically refer to tetrads as like the synaptic like complex between two homologous chromosomes. So tetrads is kind of exclusive term for meiosis, not mitosis. Because mitosis, you're just lining up every single chromosome, whereas in meiosis, you're lining up tetrads. So look, this lineup right here, these are homologous chromosomes, right? This is chromosome 1 from the mom, chromosome 1 from the dad, chromosome 10 from the mom, chromosome 10 from the dad. So we call this like tetrads, like the two homologous chromosomes together. So tetrads, just think about meiosis instead of mitosis for that.
Okay. Would that be chromosomes for mitosis then? Would that be chromosomes for mitosis, but tetrads for meiosis, right?
Yes, yeah, you just call meiosis the tetrads because when they're lined up like this. For metaphase sorry, mitosis, just call them chromosomes. They're just chromosomes lined up. There's no like, specific term for that. Okay, I'm gonna pause this video. I'm gonna try to find someone as a charger in the room. Now we're gonna talk about body systems, right? Like the actual reproductive system. And so first we're gonna talk about the male reproductive system. So the what's it called? The male sex organ is the testes, right? And then for women, it's the ovaries. So the testes, they have two like components to them. We have seminiferous tubules plus Sertoli cells. This is like one component. And then the other is going to be called, they're called Leydig cells, but they're interstitial cells. So sometimes you see them as this interstitial cells of Leydig. The reason I'm like emphasizing this interstitial stuff is because like if you get a question asking like where the locations of these things are, like okay, I don't know this for sure, but like look up a diagram. I'm sure like the Sertolis and stuff are like inside the testes, and then the Leydig cells are like outside because it's interstitial. This might be on the section bank. I'm gonna spoil that, but I'm just saying. So, anyways, what do these two things do? These seminiferous tubules and Sertoli cells deal with spermatogenesis. So, genesis of sperm, right? And what stimulates it to produce sperm? There's a hormone called FSH, follicle stimulating hormone. This is what this is. Follicle stimulating hormone in men is going to lead to spermatogenesis. Now for interstitial cells of Leydig, this one mainly deals with like producing androgens. So the best example of an androgen is testosterone. I don't know what else there is, like DHT, which is a type of testosterone, but mainly testosterone. Androgens are just like the sex hormones, okay? And what stimulates this to produce testosterone? Another hormone called LH. LH is luteinizing hormone. And what releases FSH and LH is the anterior pituitary. Okay, you know what? I'll just do it. Okay. I'm gonna revisit this in the endocrine system. But here's how it works. You have the hypothalamus in the brain, right? The hypothalamus is going to release something called GnRH, gonadotropin releasing hormone. When this releases, it's going to visit the anterior pituitary and stimulate it to release FSH and LH. And then FSH will come here, lead to spermatogenesis, and LH will come here, lead to testosterone production. So what is the pathway of sperm from when it gets produced to when it gets released? Let's write it out. I'm gonna continue it here, okay? So it goes from ejaculatory duct to here. All right, how did I just draw this out of thin air? How did I remember this? There's a mnemonic we use for this. The mnemonic is gonna be 7 up, okay? And there's a parentheses around the N because there's nothing that starts with an N here. But look, SEVE, and then once again, there's no N, and then UP. So seve with an N, so 7 up. That's how you remember the pathway. Okay, so here's the thing, right? There's 2 terms, or technically 3, I need to clarify here. Sperm and seminal, okay, well, there's semen, and there's also seminal fluid. This is giving me PTSD because I remember I was confused on this too. We had some like bio workshop where it asked us literally like what's the difference between sperm and seminal fluid and all that. And I think I got it right, but I was asking my teacher a clarification question. And I'm like I'm a junior in college, so I'm really mature, right? And so I go up to him and I start thinking, if I laugh, it's over. And so I went up to him and I asked him, so sperm and seminal, all that stuff, penis, all these words. And because I knew like not to laugh, I almost burst out laughing. And it was bad. This teacher is like a bio 2 professor. Like I was like, it was bad. But somehow I didn't laugh like fully, and there was someone else there. So you know what I'm saying? It was crazy. So I'm not gonna laugh today, but like if I laugh, it's over. You know what I'm saying? So sperm is the actual like this stuff, right? The actual cells, the the what do you call it? Gametes, right? The specific cells, okay? But when we talk about semen, if you think about like okay, when someone ejaculates, right? You have like this my fault. You have this like fluid, it's like a clear fluid, but the clear fluid, it obviously includes semen, sperm in it, but there's other things inside of it, which is the seminal fluid. So seminal fluid plus sperm gives us semen. So once again, sperm is just gonna be the cells, right? But seminal fluid is the like medium that allows the sperm to live. So it's gonna be like an alkaline medium. It contains nutrients for the sperm so they can survive. By the way, what do sperm use as a source of energy? Yes. So normally like our human cells, we use glucose for energy, but sperm in particular, they use fructose for energy. Okay. Then once again, semen is the combination of this. So the actual sperm cells are developed here, the seminiferous and Sertoli. That's what I mean by spermatogenesis. But seminal vesicles will lead to the overall combination of sperm and seminal fluid to make semen. So you have your semen here, right? Then it goes to the epididymis. Epididymis is also very, very important because epididymis is where the sperm mature. I had a question like this on my Bio 2 exam where it asked, where do sperm mature? And you know what I put on it? Prostate gland. That's like the worst possible choice you can ever choose because glands secrete things, right? So I was choosing between epididymis and seminal vesicles, and I chose prostate gland. And then this showed up on an FL, and I got the same wrong. It was so bad. So don't make this mistake, okay? Epididymis is where sperm mature. What do I mean by mature? So if you look at this sperm, I'm gonna draw a better sperm later on when I talk about the components. But this part right here, the flagella, is what allows for it to have motility, to move around. So the epididymis is where sperm gain motility, and they just chill there. And then next you go to the vas deferens. The vas deferens is like yo, what the fuck? Okay, vas deferens is where the testes get stop, bro. Vas deferens is what lifts and drops the testes, right? So hold on. Okay, so when it's cold, right, the testes will lift up, and then if it's hot, hold on. Let me just write it out. Yeah, so like the testes, they hang like below the body because they don't want to be like fully heated, right, by the body. So they have like their own like area where they don't have, it's not too hot or too cold. But when it's like cold outside, right, you don't want your sperm to be too far away from the body. And so the vas deferens will help lift it up and bring it closer to the body. and the body heat will give it heat, right, in the cold weathers. And then the opposite, if it's hot outside, then it doesn't want extra heat from the body, so it's just going to drop it. Now, the ejaculatory duct, this is just like a pathway, right? This goes towards this duct. And then you reach the urethra and the penis. Okay, look here, urethra and penis. What else follows this path? All this stuff on top is just solely for the sperm. But urethra and penis, what else goes through those two? I have a point I'm about to make with this. So look, urine also goes through urethra and penis. So here's the thing. Urine is acidic, but I keep saying that sperm have to be in alkaline environments. So how can a sperm just go through this acidic environment? Let me write some things out. What the hell? So the bulbourethral, also known as the Cowper's gland, I know there's a better term for this, but I'm just gonna say like pre-cum, okay? Or pre-ejaculate. The whole point of the pre-cum is to clean out the urethra and penis, right? From the acidic environment that was, established by the urine. So this is a function of the bulbourethral gland. It's going to produce, I'll be more scientific and call it pre-ejaculate, okay? Now there's two more glands. One I was just mentioning, the prostate gland. And then it's not really a gland, but the seminal vesicles. I mentioned this earlier, but these are what's going to actually produce the seminal fluid that the sperm can chill in, right? And the difference between these two, they're not really going to ask you this, but I think prostate gland, all it does is give a little bit of that alkaline, medium. It just kind of makes the medium alkaline, but like not to that much of an extent. Seminal vesicles will make the medium alkaline, and will also provide the nutrients and energy sources.
So are seminal vesicles the same thing as like your? I think it's also like the seminiferous tubule.
This one, seminiferous tubules?
Yeah.
Okay, yeah, good question. Seminiferous tubules are just going to make the sperm, the cells right here, right? The sperm
Right.
cells. But seminal vesicles are going to make the seminal fluid and kind of like overall turn into semen. So seminal fluid is a fluid that has
Okay.
the nutrients, energy, alkaline environment.
Right.
It's like a liquid. But the sperm are just cells. So this is going to help produce the liquid that the cells can reside in.
Okay. Okay.
All right. Okay, let's talk about a sperm cell. Okay, so this thing overall is called the head, and this will contain, draw like right here. The head contains the nucleus. This part right here is called the midpiece. The midpiece is where the mitochondria is. So you can remember midpiece and mitochondria both start with M, midpiece mitochondria. And then this tail is the flagella. And remember, where does the sperm cell really gain the flagella and the ability to move? Is going to be in the epididymis. And then up here, this little like tip part is called the acrosome. The acrosome is important because it contains the enzymes, right? When a sperm has to interact with an egg, it has to like penetrate the egg, right? So this sperm has the enzymes in the acrosome, which is the tip of the head, that allow it to do that, right? Now there's a bunch of terms: spermatogonia, spermatocyte, spermatid, spermatozoa. What are all of those? Let's talk about them now. So I guess the word for it is like primordial germ cell. That's like the original, like what you start out with. You start out with a spermatogonium. and then it's goni if it's plural. So you're starting out with this. This is 2N is diploid. Now this is gonna undergo mitosis and produce primary spermatocyte. Look, mitosis. So it's gonna be 2N or 1N? Yes. 2N. This is a primary, by the way, the one with the circle, right? And now, obviously, like there might be more cells, but I'm just drawing like a single one, right? So now we're gonna undergo meiosis 1. Now, what is it, 2N or is it still 2N or 1N? Yes, it's gonna be 1N. And then meiosis 2 is going to produce, we call it spermatid, okay? There's 4 of them because, you know, meiosis 1, or meiosis in general leads to 4. So I only drew like a single cell, but there's gonna be 4 of these. These are called spermatid, okay, with the D. And everything below is 1N, okay? Spermatids. And then when they mature, where do they mature? What did you say? Like what structure do they mature in is gonna be an epididymis, okay? So when they mature in the epididymis, then they gain the tail. So this is like your final thing, right? And we call these spermatozoa. So as you can see, they're all different things. We hear all these names. They're usually, I don't know, people use them interchangeably, but they're definitely not interchangeable. Okay, I think that's it for male. Let's see. My least favorite part. Everybody good with this? Like, can I erase it? So funny thing, right? I'm gonna be talking about the female reproductive system next. And on my Bio 2 exam, when we were learning all this, I got everything correct on the female reproductive system. But I got so many wrong on the male reproductive system. It should have been the other way around, but I don't know. Because the female reproductive system is more confusing. But I don't know. I was just being weird. I was being like, I don't know. So, okay, let me erase this. Okay. I'm so sorry. This is acrosome. Let me write this one more time. All right. Female reproductive system. What time is it? Okay. So I'm gonna draw an a uterus. Crazy. I don't even. Let's see if I can draw it. It's like something like that. You guys know what a uterus looks like, right? Like the whole. No, you don't. Okay, something like this. So I'll label it. This is horrible, but basically, this area inside is like the main. This is how it looks.
Wait, are you including like the? Isn't there like a fallopian?
This is,
tube?
bro, let me explain, dog. So this part right here is like the main thing that we refer to as the uterus. This little like I don't know, like circular tube thing is the fallopian tube, or it's called oviduct as well. And then there's 2, right? Now this circle at the end is the actual ovary. There's 2 of them. And as I was mentioning, the testes are the sex organs of males, and then the ovaries are the sex organs of females. Now you have like this thing that gets produced, which is the endometrium or endometrial lining. This is what gets bled out when women have periods, right? You establish this endometrium and it gets bled out. I'll elaborate on this when I get to it, but I'm just I'm drawing it just to show the structures of everything. And then this tapers like this. And so like when you reach like over here, this ends up being like the vagina. It's like the, you know, the outer part. And then the area right between that like separates the uterus from the vagina is called the cervix. So just some like terms there. Okay, so now let me explain some terms, okay? So we have, as we had like sperm cells, we have egg cells, but there's also, I mean, they're called like, the eggs are going to be called oogonium or oocytes. I'm going to do this again with the eggs, but I'm just saying it's going to be something, right? But there's also something called a follicle. So a follicle is like I don't know, like an outer coating that eggs will reside in and develop, okay? And then the follicle is what ends up rupturing later on, leading to the release of egg. And this is called ovulation, when the egg gets released. But this is what a follicle is. It's just something that like kind of helps nourish the egg while it's developing, and then it does a bunch of other stuff. I'll have to get to that to mention, but I'm just trying to define some terms. So here's the thing, right? Men, right? We produce sperm in puberty, okay? Nothing before that. But for women, they produce all of their eggs before they're even born. So while they're like still fetuses. Yeah, okay. So here's what we're gonna have. We're gonna have a oogonium, which is 2N. And this is going to do lots of mitosis and just keep replicating itself. You're making all these oogonia. And then at some point when you're done, it's going to differentiate into a primary oocyte. Now I'm gonna draw this line right here. This primary oocyte, remember this line refers to before birth, okay? This primary oocyte is arrested in prophase 1 of meiosis. And this is 2N. What does arrested mean? Like when I say it's arrested in prophase 1, yeah. So it's just stuck there in prophase 1 before birth. Now, you know, you made all of these. If you made like 20 eggs before you were born, then that's all you're gonna have to ovulate in your lifetime. If you made 100 eggs, then you ovulate all of those, okay? But all of this is determined before you're actually born. Now, the period between when you're born and puberty, nothing's gonna happen. But when puberty hits, this, I don't want to, okay, I'm just gonna continue this, okay? So puberty, now it's going to finish off meiosis 1, okay? So now you're producing a secondary oocyte. And there's also, you know how meiosis will create, it will create 2 cells per cell, right? So there's 4, but I'm just drawing 2. So you're gonna make a secondary oocyte, which is 1N. And you're making I'm making this really small, okay? This is called a primary polar body. So the thing is, we don't really know what the purpose of a polar body is, but like the way it works is that polar body and oocyte, they'll have the same DNA, they'll have the same genetic material, but the secondary oocyte took all of the cytoplasm, all of the organelles, and everything else. The only thing this primary polar body has is very little cytoplasm, and then like, you know, the genetic material. But the polar bodies that we produce, they just get like, I don't know, like recycled, thrown away. But this is the main aspect of this whole thing, secondary oocyte. Now the secondary oocyte also gets arrested in metaphase 2. And it gets arrested there until a sperm will fertilize it, okay? So I'm gonna just leave this here for now. Now, to kind of like go alongside this, the whole follicle thing I was talking about, you just have like a primordial follicle, and then, I don't know, it just develops. I don't know if there's proper names for this. and it just develops, okay? But the egg is going to be residing in the follicle and developing like this while the follicle is developing, okay? Now, at some point, right, the term is ovulation, okay? Ovulation just means the egg gets released from the follicle. So it's going to look like this right here. Here's your egg. Now, this follicle, now when it's released, the egg, this becomes something called a corpus luteum. I'm just going to draw it like this. Just to add to the diagram, if a sperm comes, then this will continue meiosis 2 and then form a zygote, which is 2N. Why is it 2N? Because the sperm is 23 chromosomes, the secondary oocyte is 23 chromosomes, so they come together and form the zygote. But anyways, a corpus luteum is there after the egg gets released. And the corpus luteum, if the egg gets fertilized, it's going to remain. But if the egg does not get fertilized, then eventually this corpus luteum turns into something called a corpus albicans. I'm going to draw it as a square. And it's just like a non-functional corpus luteum. It's just useless. And what do I mean by functional? Corpus luteum, the function of it is to... Progesterone. Yes. This will produce progesterone. Now, I know this is really confusing, so I'm going to connect everything together. So firstly, let's talk about the enzyme, or what's it called, hormones. And then I'll put it all together, progesterone, all that. So we have FSH and LH again. What is FSH? Follicle stimulating hormone, and LH is luteinizing hormone. So follicle stimulating hormone, it's just going to stimulate a follicle to be, we have one of these follicles, they're all just sitting there. One of the follicles will be like, not like released, but like starting to get developed, right? So this is what FSH does. LH, luteinizing hormone, this is mainly important for this right here, ovulation. LH is going to lead to ovulation. And you can kind of connect it, right? Luteinizing and corpus luteum. So they're connected like that, right? One second, I'm super dehydrated. Okay. Now, what is estrogen? Estrogen, like we always think of it as the opposite of testosterone, right? Both testosterone and estrogen, they'll lead to like secondary and primary like characteristics, sex characteristics, right? So what I mean by like secondary for men, right? Testosterone is going to lead to like body hair and like deeper voice, Adam's apple, all that stuff. Estrogen, some secondary sex characteristics are like I don't know, wider hips. But anyways, estrogen, that's like the thing that it does. But the main thing that connects to all of this is going to be that it establishes the endometrium. So looking back at this, right, the endometrium is there because the plan is that there's a fertilized egg, and this is like a nice environment for it to develop. So that's what estrogen does. Now, progesterone will protect the endometrium. So look, E estrogen, E establish, P progesterone, P protect, or you can even do PR and ES. So like this estrogen will just like be there to develop all this. But then as we were talking about, right, this corpus luteum, when the egg gets released, the corpus luteum is there for a little bit and it produces the progesterone regardless of fertilization, right? Because it's assuming that the egg will get fertilized. And so it's going to develop or secrete progesterone so it can maintain this. And then if nothing occurs, no fertilization, then your secondary oocyte is just gone, right? And then your corpus luteum becomes this corpus albicans, and then your endometrium just goes away. That's what periods are, right? You're just bleeding out, the endometrium breaking down. You know what's funny? Okay, my sister is 11 years old, right? And so she's learning about periods now because she's in middle school. And so I asked her, what are periods? And she said, oh, it's like the thing when the baby thing bleeds out. And then she didn't really know for sure, right? So you know what I told her? I said that women are inherently bad. And so God made it so that they bleed out their bad blood every month. So that if they didn't bleed out, then they would become like monsters. And she was listening. She was just like, oh, that makes sense. Oh man, it's crazy. So, anyways, now let's talk about the menstrual cycle, the worst part. Here's the thing: if you have like a device on you, pull up the menstrual cycle like diagram, because like I'm gonna draw it out, but it's better if you follow along. Let me 1 second. Yeah, for anyone like watching like online, it looks like this right here. It's like this cycle where it has all the hormones and all that. I'm gonna like explain it, of course, but it's easier if you have it pulled up. Okay. So let's start over here. The beginning is gonna look like this. So in black, I'm gonna draw the endometrium, like thickness and all that, right? So it's really thick here, and then it drops, and then it redevelops and all that. So in the beginning, like the first few days, the endometrium is really, really thick. That's just the start of the menstrual cycle is when the blood comes out, right? When the endometrium breaks down. That's just how we refer to it as. And at this point, estrogen and progesterone are going to be low. Fuck, bro. Okay. In red, I'll refer to it as estrogen and progesterone, and I'll specify if I need to. So estrogen and all that is low, and then here, something like that. It's bad, but it's something like this. Okay, just follow along with your diagram. And then your FSH and LH are going to be like kind of high, and then at some point it goes like that. Okay. So I'm referring to purple as FSH and LH, but in particular, this peak right here is your LH. And then estrogen also kind of goes up, okay? So remember, this endometrium broke down because there's no estrogen, which establishes endometrium, and there's no progesterone, which protects the endometrium. Now this is the start of a new cycle, right? Your endometrium is gone, your follicle is gone, whatever. So now it restarts. You're forming a new follicle. You're getting ready for it to develop, and hopefully fertilization will occur. So that's why I know this is really poorly drawn, but that's kind of how the diagram shows it. But like your FSH and LH are like kind of like increasing and stuff for that purpose, right? What does FSH do? It nourishes the follicle, and then LH is just there, but it's not going to do anything until this peak. This peak is the luteinizing hormone, right? Where it causes ovulation. This is what I was referring to earlier. As you can see, estrogen, let me draw it better. Estrogen is also like really high, and then whatever. And then after this, your estrogen and this is okay. Your progesterone is going to be a little higher. Estrogen is kind of lower, and then FSH and LH are just like kind of there. But let me break this down. All right. So we talked about this ovulation and estrogen. Okay. So here's the thing. This is really confusing. This is what I was referring to in the very beginning. That it's just annoying to explain and learn. But let me give you the main takeaways from this. A big thing that both like college and even MCAT care about is feedback loops. So estrogen, a lot of things that we have are negative feedback, right? So estrogen, when it's let's say like medium, right? Medium amounts, not like crazy high, but medium amounts. This is a negative inhibitor of FSH, LH, and then GnRH, right? GnRH is the hypothalamus hormone that leads to the release of these two from the anterior pituitary. And why does this occur? Because estrogen is establishing an endometrium, right? And yes, you want some FSH in the beginning to get that initial follicle to be stimulated to even be developed. But while you're like establishing endometrium and all that, you don't want to overstimulate FSH, because what's going to happen if you have a lot of FSH? You're going to be developing too many follicles. We only want one follicle at a time. And same with LH. You don't want to like have all these follicles that all get ovulated. That's not how it works. You just want one at a time. Now, when you reach this point, estrogen, as you notice, right? Estrogen and LH are high. So estrogen at some point, this is like day 14 or something. Estrogen, when it's high, it actually, let me write this in a green color. It positively feeds back on those. So you're gonna have, of course, like FSH as well, but the main thing that gets activated is your LH. So I'll put some FSH, but mainly LH. Your estrogen peaks here because it's, finally establishing all this endometrium, and it's ready for the egg to get released. So it's going to temporarily cause luteinizing hormone to spike up, and that will be just enough to cause the egg to release. And then after that, your LH and FSH are like kind of low, and so are your progesterone and all that. But now look what happens, right? The LH caused ovulation to occur, and it also stimulates the corpus luteum to secrete progesterone. So LH right here, that. But as I was saying, LH is going to spike up and it's going to go down because, once again, we don't want to ovulate all these different eggs. So what happens? How do we get corpus luteum to secrete more progesterone? Do you know how pregnancy tests work? What is a pregnancy test like check for? Yeah. Something new that gets produced after fertilization is HCG. So we're assuming that a egg got fertilized, right? And so now we want this corpus luteum to stay, and we want it to continue producing progesterone. So instead of LH, right, this is gone. We don't want too much of that. We have HCG. This is human chorionic gonadotropin. And this is just an analog of LH. Analog means it's very, very similar in structure, but it's not the same thing. So HCG is similar enough to stimulate the corpus luteum to secrete progesterone. And once again, we need this because progesterone is what maintains, right? It protects the endometrium. That's it.
So it seems like ACG is only released once like the sperm kind of binds onto the
Yes.
egg.
Yeah, because HCG, right? It's there so that the corpus luteum is still there. We don't want it to go away. We want it to stay there and produce more progesterone. In normal cases where there's no fertilization, this corpus luteum will stay there for however long, like a couple of days or whatever, and the LH will be just enough to allow it to survive for a little bit. But then, since there's no fertilization, this HCG is not there. LH dies on its own, and so the corpus luteum becomes a corpus albicans, it dies, and no progesterone. Then this endometrium is not going to be protected. Why can't I speak, bro? It can be protected, and then blood, right, from the endometrium breaking down.
So nothing is really like binding onto the endometrium at this point whenever HCG is being secreted.
Yeah, not the endometrium at this point. Actually, thank you for bringing that up. I need to mention one more thing. So we have our fallopian tubes, right? The egg gets like produced here and then it releases. The egg gets fertilized in the fallopian tubes, okay? So that means that the sperm, one second. I'm gonna call it the oviduct, okay? Same thing. So the sperm, it has to go through all of this, right? And end up in either of them, okay? That's why, as I was talking about before, the seminal fluid, the alkaline environment with nutrients and all that is necessary for sperm so that it can survive the entire pathway through the male and all the way into the female. Because I don't know what type of stuff is going on, like if it's also acidic, that's like the idea. That's why we need alkaline environments that can survive all the way up until this point. And then don't forget the sperm has the acrosome, which is the tip of the head, and then it can penetrate the egg. We're going to talk about more about fertilization in next chapter, like tomorrow. But okay, look, this will also be mentioned tomorrow, but really quick in case anyone's confused. Fertilization occurs here in the fallopian tubes, but after a couple of days of the zygote and the embryo developing, then it moves to here, okay, and it implants into the endometrium and develops there. Do you know what it's called if the embryo does not implant here and just stays either in the fallopian tube or somewhere where it's not supposed to be? Yes, good job. That's called an ectopic pregnancy if the embryo is not where it's supposed to be, which is the endometrium. This is funny because I in my EMT class we were learning about ectopic pregnancies, and like I'm sure they're rare, but I had one clinical like right after I learned about it, and then the it was pretty boring day, like not much was going on, and then everyone was rushing to one room, and I was like, "Yo, what's going on here?" And they said this lady's having ectopic pregnancy, so I was like, "That's pretty interesting." And it was crazy. They had to like I don't know, her like stomach was like inflated. They had to like pump a lot of blood because what do you think happens if the egg is or what's it called the embryo is developing in something as thin as this? What's going to happen? Yeah, yeah, this thing will rupture and then blood loss. That's what was happening to the lady, and they had to like do some like transfusion, crazy stuff. I didn't even understand it, but yeah, I was surprised to even witness an ectopic pregnancy. But yeah, you don't have to know ectopic pregnancies for MCAT. It's just like fun facts or fun information.
It's called the ampulla or something.
It's actually very common.
Is it actually?
Yeah, I'm a nurse and we see that all the time. So
Okay, that's cool. Maybe that's why I saw it. Because the place I was at, nothing crazy ever happened. So that's cool. What were you saying, ampulla?
Yeah, I think that's the specific part of the fallopian tube where like fertilization happens.
Yeah, I mean, I don't know if you have to know all that for the
Oh,
but I'll just mention
yeah.
it. Like, yeah, this is the fallopian tube. You have the egg starts here, and then you have these little hair things called fimbriae. These are little hairs that push the egg up, or in either case, and then the egg will chill in the. Okay, I didn't draw it in such a way, but like the widest part of the fallopian tube is called the ampulla, so like AMPULLA, and that's where the egg will chill and get fertilized, and then eventually it will come down here. All right. I don't think there's anything else. The only thing I can think of is like, okay, really quick. The I think I'll mention this tomorrow as well, but the egg is like gigantic, and the sperm comparatively is pretty small. Okay, so both of them have half DNA. This one has the 23 chromosomes. This has 23 chromosomes. But the egg is going to contain all of the everything else, right? Cytoplasm, organelles, everything like that. Which is why whenever you deal with genetics questions or you see in a passage, they mention mtDNA. What is mtDNA? Mitochondrial DNA. I mentioned this chapter 1 as well. The mitochondria, chloroplast have their own DNA. Chloroplast is in plants, so it's irrelevant. But mitochondria have their own DNA. Whenever we talk about mitochondrial DNA, that could have only come from the mother because the egg came from the mother, and this contributed all of the organelles. What is the organelle? Mitochondria is one of the organelles. So for dealing with mitochondrial DNA, it came from the egg, from the mother. But don't forget, they have equal amounts of genetic information. And then this last part, I don't know how necessary it is. It's just like the phases of the menstrual cycle. Yeah, so like, I don't know if I mentioned it. This is the part I was talking about in the beginning where I got like confused because everything overlaps.
Follicular phase, and then your algae surge, and then your
Yeah, so
luteal phase.
it's weird. There's like multiple different phases, right? So the first half is like the follicular phase where your follicle is developing and all that.
0 to 14 days.
Yeah, because I know like a month is 30 days, but like we kind of refer to it in 28 days. So 0 to 14 is your follicular phase, and then like technically 15ish, like 14 to 28 is going to be your luteal phase. This is pretty easy to remember because this is the follicular phase, right, where the follicle is developing. And then after the LH spike, where luteinization occurs, that's just the ovulation, the egg gets released. This is the luteal phase. But this is the part where I'm really annoyed. So this is 0 to 14. From 0 to 5 days, generally, this is called menses, which is pretty much all about like the endometrium coming out as blood, right? And then day 6 to 13 is the proliferation phase. So like you're really developing that follicle. And then 14 we know is the LH surge, right? Egg is released. And then day 15 to 28 is the secretory phase. Yeah, once again, I don't think you have to like know these specifically. It might help you, but like clearly I didn't remember it. But once again, right, the main things to understand is like the cycle of like hormones, and that I guess the main takeaway, right, is estrogen. Okay, estrogen, it can do both positive and negative feedback. And it does positive feedback if it's really, really elevated levels, but it does negative feedback if it's below that, so medium and below.
So, when you say menses, so
Yes.
does menstruation happen first?
Yeah, it's like, yeah.
Your, like your ovulation, or?
Yeah, so based on the way like we what's the word? Oh, conventionally describe it, we start the cycle with menstruation. I know this seems
With like shed.
yeah.
Okay.
That's what this black stuff is, the endometrial thickness. So it's really high on day 0 and then it gradually becomes lower and then it's redeveloping.
Okay.
Yeah. And while the endometrium is being shed, now you're developing a new follicle, which is why FSH and LH are relatively higher than later on. All right, a lot of information between this and the very beginning. Like we talked about cellular methods of replication as well as like the actual reproduction system. So anybody have any questions on anything? It could be this or just anything I talked about in the beginning.